All right, thank you.
You're on.
So when we have, of course, when we have established all the type of tests that as a laboratory we want to offer, for instance, we have also to talk to the clinicians, and the clinicians have to determine, clinicians or healthcare providers like genetic counselors, they have to determine what type of test is the most appropriate for a particular patient. So if you have a patient that underwent a very firm diagnosis, then looking at the entire genome may not be the first choice, so the first tier. But the first tier will be a more targeted approach where you analyze all the genes that have been associated with that particular diagnosis. Usually, in addition to a diagnosis, there is also a differential diagnosis that can be useful. So the physicians, they want to exclude other diagnoses. And that's why gene panels have been put in place in order not only to have the genes that can cause a particular disease, but also genes that can be associated with the form first of that disease or alternative presentation of that particular disease, or diseases that are similar. Just to give you an example, if you have a patient with an aortic dissection, not always the cause could be fibrillin 1 for Marfan syndrome, but there are other diseases like Loeys-Dietz or the vascular Ehlers-Danlos disease, which can present with aortic dissection, dilation or dissection. Therefore, you want to have, when there is a patient with an aortic dilation or dissection, then the physician will be best in ordering a panel that has all those genes that can be associated with that trait, that specific phenotype, which can be part of multiple large syndrome. The advantage of having gene panels is that having a lower target, or a smaller target rather, you can have a much higher coverage. Much higher average coverage means a much higher quality for each data point, which means a better quality overall and of detection of all the possible changes that occur in the targeted region. And also detecting variants in genes for which there are a lot of knowledge, there is a lot of knowledge already available, there is a lot of information, and even this makes the classification, clinical classification from the laboratory much easier. So better metrics can lead also to a better sensitivity and specificity. And it fits better with the current reimbursement model for diagnostic tests. So insurance companies are much more inclined to accept a testing from a genetic panel, gene panel, rather than going straight to exome or gene.
Do you think at one point that it will be a requirement for every newborn to have their genetic testing done, and then the insurance company doesn't have to pay it again and again?
Yes, I think that we will reach that point probably. Keep in mind that, the nuclear genome, it's not going to change much during our life.
Right.
Our epigenome will change dramatically during our life.
None of the current tests, I mean, especially
no. What it means is that there will be one day in which every newborn will have not only the sequence done, complete CMV analysis, epigenome done as well, and everything so can be done with probably one assay. Yeah. So this will be the fulfillment of some requirements that probably will be in the future. Right now,
I think it's cheaper for the insurance company, right?
Well, yeah. The thing is that we don't have. I mean, it's cheaper to have one data and data set, and then going back to this data set, if nothing has been that current, according to the current knowledge, nothing has been found of interest, it can be you can go back and analyze with new tools, new knowledge, and see if there is something there. The problem is that not always everything is in just the sequence, and so that's where, but I think it's going to be cheaper. Probably at the end of this year, doing genome sequencing is going to be much cheaper than most of the panels. So what happens if we don't have an answer for a panel, or the presentation of a patient is quite complex, or there are a lot of confounder factors? I've seen myself in patients with exome sequencing done that they had two variants causing two different diseases simultaneously. And in fact, when you then go back and compare the clinical presentation, these make sense, how it was confounding, and therefore the clinicians, they were thinking about one disease, but they were thinking also another disease, and they could not make a definitive diagnosis in those patients, and therefore they sent an exome rather than a panel. The exome always keep in mind that it is not covering the entire coding sequence. We have places that are missed because of the capturing is not perfect, and always requires Sanger, of course, Sanger confirmation, because the coverage is usually lower than the gene panels. If you're not satisfied with what the exome can give you, you can always go to the genome. And the genome usually are low coverage, between 30 and 40x coverage that has been used in the past, but we know that is insufficient. So it's low coverage. It's not feasible to cover parts that are not covered well by the genome, even though the genome does not require the capturing, the enrichment, but you still have part of the genomes which are highly repetitive. So chromosome 6, for instance, has histocompatibility complex genes, which are really quite messy. And so, and the thing is that not always it's easy to interpret non-coding variant. So if you have a deep intronic change that is not in those places that I mentioned before, then it's difficult to make an interpretation. So what happens when the massive parallel sequencing is inconclusive? So you have negative panel, negative exome, negative genome sequencing. So, you don't have suggestive US, or, the MPS data do not support the inheritance model that you have generated for that particular patient. Then, can a second hit be present, but, was not detected, for instance, in the exome sequencing, and it was something that was in an regulatory sequence, in an enhancer on a silencer very far from, or even for a genome sequencing where you have all the sequences, but nothing come up of interest. So it could be large indels. So in the past, the elements and the events that leads to large deletions or duplications have been tackled by alternative approaches like microarray analysis, or MLPA, which is a multiple ligation probe amplification. And but those technologies, they have their own limitations. So first, the CMA, you don't have all the probes in all the places, and the CMA cannot really detect balance translocation. They cannot detect inversions in the same way unless it occurs right on the probe that have been placed. MLPA, which covers each exon, only they provide one probe per exon, And so if you have a partial deletion or a partial duplication occurring in a spot where the probe is not there, the probe will not detect that event. So can massive parallel sequencing data help? Of course it can, and it does. So the all stands on the how much coverage we are able to obtain, and how this coverage and how many samples we can run simultaneously. Because even though the coverage is not exactly the same, but is reproducible, meaning that if you have 10,000 samples, the amount of coverage will be similar in the same positions. Not equal, but very similar to the same position. So if you have a difference that occurs in coverage, where you can have either more coverage than what you should expect in a position, you can have duplication. Or if you have, a deletion, both in heterozygosity or in homozygosity. So, and this has been very nicely identified also with the split read analysis type, where you match everything to the reference sequence, but then you have places where this reference sequence does not really match and you have a messy signal, but you can have a split read both at the 5' and the 3', which can entail a deletion of an entire exon also. So you can create a quantitative read plots where really you can detect whether there is a half amount of signal of coverage that you would expect for a certain gene. So this is an autosome. Of course, this is a KCNH2, is a potassium channel gene. So you are expecting 2 copies if this has a normal amount of alleles. But in this case, you see that there is a deletion on exon 3, and with the split reads, these turn out to be a partial deletion affecting intron 2 and exon 3. But here comes, even more fun, is that with split read analysis, it was identified that actually not only there was a partial deletion, but there was something weird going on, and there was essentially an inversion of the signal from the other exons. So in this case, it was detected not only a partial deletion of exon 3, but also the inversion of the remainder of the gene. So this is a replication-based type of mechanism. Could be FOSTIS or MMBR. FOSTIS is fork stalling and template switching type of where, so during replication, the replication machinery stops working and detaches and then reattaches to the place where the machinery thinks they left, but actually is a homologous place, very similar, but not the same. So this type of event, this type of complex rearrangement can be detected by next generation sequencing.
You wouldn't be able to do that with the whole exome sequence, right? So this can only be done for whole genome.
This actually was done with the gene panels.
How can you do that?
The way I explained, so with the split read.
Can you go back?
Yeah.
Now, so for this one, I understand, but if we go to the next one, with the kind of the inversion ones, you cannot, right? If you only do the whole exome sequence, like this.
No, I can tell you this was detected. Yeah.
But in that
but this was confirmed by Paco Bioscience reading.
Right. So
yeah.
In that case, you have to be. It's very low sensitivity because the split point needs to be very close to the exons.
That's why you need yes. That's why you need also to confirm by another technology.
Well, I'm not. The specificity is not something I worry about. If you see that from, it's in terms of sensitivity.
Oh yeah. Depending where the probes are placed. Yeah.
I mean,
yeah.
1 out of 1,000, you probably can identify this because the junction point, the split point is within axon or close to axon. It's very, very low.
Yes, depends very much on where you put your probes.
Right.
Yes.
So
if you put your probes very close to the exons, the vast majority of you won't see it.
Right.
But in this particular case, I can tell you that the probes are covering extremely well the entire region, even the intronic region. So it has to be super deep intronic in order to be missed in this case.
But you have to be really lucky to do this, right? Or your panel is designed to detect it.
Correct.
Yeah.
This is the last. Yeah.
In most cases, the panel is not designed for this.
No. You have to design in a way that you are able to detect it. Yeah, that's correct. And this will be much more, much better identified by genome than by exome. Exome will miss it because you cannot afford to have so many, baits.
So what do you call it? Is it an array then?
No, this is next gen.
So
this is
next gen, but is it like a glass slide with lots of probes?
This is using an Illumina machine, same machine that Yulong has.
So the impression is the panel design, they will design the probes almost tile the entire gene for this particular purpose.
Correct. Correct.
So you're able to identify the real junction point.
Correct. Right. And it goes even beyond, part of the 5' and 3'.
Sure.
Yeah.
Can you quickly go over, like, you have a nomenclature of this slide?
Yeah. Oops.
How do you read them?
Well, it's, you have the deletion part here and the inversion of the last part of the, so there is deletion, insertion, because of course this is deleted here, but inserted here and inverted. So this is inserted and deleted, inserted and inverted. Does it make sense? Meaning you have a partial deletion, the remainder was gone and has been reinserted opposite. So it's inverted.
It's complicated.
Yeah.
This is for cytogenetic. But in any case, this is this system has been proven to be way more sensitive than the regular microarray. Of course, if you design microarray in a different way, but then microarray analysis, so
very difficult.
So
maybe PCR is fine with no very junction point. But this one is you can identify the de novo.
Right. This was de novo. Yes. And so the advantage of that using massive parallel sequencing is that you can have less noise, more even coverage depending on where you put the probes and how you use also anti-probes in order to even up the signal, most accurate detection of CMV breakpoints depending on, again, where the probes are. But especially for genome sequencing, the CMV breakpoints is something that is very easily obtainable and can detect also low-level mosaicism if you have the right level of coverage, and can detect the balance rearrangements, which is usually missed by CMA. So looking further, of course, we have started with genomics, but we are getting close for epigenomics. Will be the next, the next boundary for clinical application.
How do you do the heart biopsy?
No. For instance, just to give you an example, so there are diseases that are due to hypermethylation or hypomethylation,
right,
and can be detected. Those are constitutional, and so blood sample, and you're fine. Others that, of course, may not occur constitutionally, but they are somatic, and those require the tissue.
Yeah. Okay. So but this part is it's more concentrated on you just get the blood samples and to do the methylation?
Yeah, this is what currently it's possible, and is done in clinical testing laboratories, is using genomic DNA and of course also mitochondrial DNA, but mostly it's genomic DNA. So just to some messages that take-home messages is that clinical massive parallel sequencing actually has been put in place because it's reflecting the complexity of the human genome. So where we need more information at a higher depth and a higher throughput, there are a lot of technologies that can lead to the detection and the capturing and detection of sequencing data, and there are different modalities and also bioinformatic pipelines for variant detection, calling, and curation. The clinical interpretation is, of course, the most important part of a clinical laboratory, and is based not only on type of variant, on the frequency, but those two metrics, even though they are have been always important, they may not be sufficient to yield all the evidence that you are required. Functional studies would be, of course, a plus. There are a lot of variants for which functional studies could make the difference between a not classification or VUS and classification as a pathogenic or a tolerated. And so massive parallel sequencing as the last has been a technology so pliant, so versatile, that really it's going to replace most, if not all, the application of other applications such as MLPA or arrays. And therefore, we are looking for massive parallel sequencing as a possible test which will replace a lot of other tests. So with one technology, you can have multiple applications. Questions?
Questions?
So first of all, thank you for bearing with me for 3.5 hours.
No, that's great. Thank
2.5
you.
hours. Yes.
These panels are moved by the FDA, by medical devices. They go to the vote.
FDA, so there are some FDA approved tests, yes. So there are some companies that they have developed a machine and an entire test, for instance, for CFTR, cystic fibrosis, and it's an FDA approved test. So you buy the machine from this vendor, and you will get also the reagents, and everything is FDA approved. So you don't need to undergo all the validation process that otherwise you would do, much more complex, because everything has been already validated. You need to make a validation, but it will be very, very less complex than otherwise it would take if you do a laboratory test development type of. So, and FDA, not all the tests are FDA approved, but they are all under CAP and CLIA, which is as simple as certainly the institutions that are confirming that that everything has been done by the book, according to all the guidelines, to all the standards that are accepted worldwide.
So can you comment on the regulation for the incidental findings?
Yeah. So when we do exome and genome in particular, but this can occur also in very large gene panels. Sometimes we can detect variants that are present in genes that are really not directly connected to the phenotype. Let's make an example. 22 years old female with a metabolic condition that has been going on for since she was born, and but they didn't was negative. A panel for that condition, for genes affecting that condition, was negative, and they send whole exome sequencing. So they identified some variant of uncertain significance in some genes that have preliminary evidence for that disease. But more importantly, they have identified a very well-known, variant in BRCA1. So the American College of Medical Genetics and Genomics has published guidelines to how to report and what to report as incidental findings. There are a series of, a core of 59 genes, but there are more than that, causing cardiovascular and cancer diseases that need to be, they need to be handled with care, in the sense that if they find incidental findings, so findings that were not for that purpose, they need to be reported, because they can have an immediate and important implication for the healthcare of that particular patient. So even that patient may have as a primary problem the metabolic problem, but she, in this case, bears a high susceptibility to breast cancer in her lifetime risk.
But the patient can opt out from incidental.
Yes. So when they order the test, you can opt out or opt in, meaning that I want to know or I don't want to know, and this can be done if the patient is adult or 18 years or older. There are some genes that are not, for which, depending on the age of the patient, they will not be reported, like long-term neurodegenerative, let's say, if the age of onset, late age of onset neurodegenerative disease in 2 years old with a very, very severe metabolic conditions or something like that. So there are some exceptions. There are genes that even they're discouraged to be tested in patients that are below 18 years of age, like for instance for Huntington disease, which is of course a different test. It's an expansion, and so kids are not, they're not suggested to be tested because, of course, this entails also a psychological, sequela for which the healthcare system is really not ready. I mean, this is a death sentence.
So one thing I didn't know the answer was. For these testing results, I know that this will go back to the doctors and go back to the patients potentially. Will these results go to the insurance company?
So the clinical report always go into the chart, and so the chart.
How about the incidental findings?
The incidental finding is part of the report, so it goes to the chart.
So that potentially is a problem.
Yeah, luckily, thanks to new laws like GINA, which is the Genetic Information Non-Discrimination Act, in theory, insurance companies and military personnel are excluded by GINA. But insurance companies should not treat differently for pre-existing condition or for conditions for which there is a susceptibility that has been identified. We know that, of course, what they should do and what they do in reality may be different, but that's why we have the federal government that is supposed to.
So what you are saying is different from the Affordable Care Act. that there's a pre-existing
condition.
conditions shouldn't be
N-
considered.
No, this should not.
So that is a
should
different
be.
act. That
It's a different act. Yeah, it was preceding the
Okay.
Affordable Care Act. So yeah, so the ACA was later after GINA.
So the insurance company shouldn't stop insure someone that because they have the RCA.
No, I know, but there are loopholes that they are using essentially, and yeah, this is not the place or the time to discuss
No,
healthcare.
Those regulatory.
No, meaning that it will require more than 3 hours.
That's to be discussed around the bar, right?
Other questions?
Other questions? All right. In
Thank
that
you.
case, thank you very much.
Thank you.
And everything being discussed today will be in the midterm exam. Just making sure you're aware of that.
The slides are not available.
Oh,
The slides.
yeah, it will be available. Yeah.
All right. Thank you.